Hello guys, welcome to my YouTube channel. So in this video, we are going to talk about the salvage pathway for purine nucleotide synthesis. Now, the salvage pathway is just a pathway that is going to help us recycle the nitrogenous bases that has been broken down from the nucleic acid catabolism. Now, when we break down the nucleic acids, we are going to form the individual components of the nucleic acids, right? The nitrogenous bases, the pentose sugar, and the phosphate groups. Now, I want to see how we are going to salvage or recycle these nitrogenous bases into the purine nucleotides, right? Now, this salvage pathway is very much crucial, especially in the bone and the bone marrow and the brain. The bone marrow and the brain lack the de novo pathway for the synthesis of purine nucleotides, and therefore, this is the only pathway they have so as to synthesize the purine nucleotides. This pathway also helps the body to synthesize these purine nucleotides from the bases present in the bloodstream. Now, when you break down the nucleic acids, a particular portion is broken down into uric acid, which can then be taken to the kidney releasing urine or excreted from the body. But now, in this case, about 90% of the nitrogenous bases from the catabolism of nucleic acids are not converted into uric acid, and therefore they are going to be salvaged. Now, let us see, for example, what is going to happen when you have nucleoproteins. Nucleoproteins are just like the nucleic acids bound to proteins. Now, inside the stomach, nucleoproteins are going to be broken down into two. They are going to be broken down into the individual components, the nucleic acids, and the proteins. Proteins, of course, can be acted upon by enzymes such as pepsin. Then they can be broken down into individual amino acids. Now, for the case of the nucleic acids, these nucleic acids can be acted upon by enzymes such as the ribonuclease and the deoxyribonuclease, or you can just denote them as RNase and the DNase, the ribonuclease and the deoxyribonuclease. Now, these particular enzymes are produced by the pancreas. Now, pancreas produces these enzymes. Now, when they get into the small intestine, the duodenum, they are going to be acted upon by these particular enzymes. Then you are going to form nucleotides. Because the nucleotides are the ones that bind over each other, maybe by the help of the phosphodiester bonds, the hydrogen bonds, the base stocking interaction. Then you are going to form the nucleic acids. Now, when you break the nucleic acids into its individual component, which are the nucleotides, you are going to use those particular enzymes. Now, nucleotides can in turn be broken down into nucleosides. So, nucleosides. And now, we are going to lose the phosphate group. We are going to get rid of the phosphate group. Alright. Now, the nucleoside, of course, the nucleoside. Nucleotides are just like the nucleosides which are bound to phosphate group, maybe 1, 2, or 3. So that's the only difference. Only the phosphate group. Now, nucleosides can in turn be broken down into two components. Now, this particular enzymes here are called nucleotides. So, nucleotides. Let me write this in blue so that you can have some uniformity. RNase and then DNase. Alright. Then nucleosides can be broken down by enzymes called nucleosidases. Which are going to break the nucleosides into the individual components. The individual components of nucleosides are the pentose sugars, and the nitrogenous bases. Of course, with the nitrogenous bases like the pyrimidines, the adenine, guanine, all those, thymine, cytosine, uracil. Now, we are going to have those individual components. So, these particular nitrogenous bases are going to be salvaged. Now, let us see some of the examples of salvage processes. So, for example, when you have adenine, I just told you that you can have adenine, and have guanine, maybe you can have right. Now, for example, let's say you have adenine. What we synthesize the nucleotide from this particular nitrogenous base. So adenine can be converted into AMP, that is adenosine monophosphate. Now, what is going to happen? We are going to involve the PRPP. PRPP is going to be converted into pyrophosphate. Right? And this reaction is going to be catalyzed by an enzyme called is going to be called adenine phosphoryloside transferase. Adenine phosphoryloside transferase is going to convert adenine into adenosine monophosphate. Of course, you see we already a phosphate group there. Number two, we can have maybe guanine. Guanine can be converted into GMP, that is guanosine monophosphate. And this reaction is going to involve the PRPP into pyrophosphate. PRPP is just phosphoryloside pyrophosphate, or 5-phosphoryloside 1-pyrophosphate. Then this reaction is going to be catalyzed by an enzyme called guanine or hypoxanthine. Guanine phosphoryloside transferase. Right. So that particular enzyme is going to convert guanine into GMP, which is a nucleotide of guanine. Then, actually, this salvage pathway is very, very much important. And this enzyme, the hypoxanthine guanine phosphoryloside transferase, is the most important enzyme in this particular pathway because guanine is mostly released in the form of hypoxanthine. So let us see how we can convert this hypoxanthine. When you have hypoxanthine can be converted into inosine monophosphate, or just IN. Alright. Then this reaction is going to be catalyzed by the same enzyme. We can have PP, then have the pyrophosphate. Then the enzyme is hypoxanthine, guanine, phosphoryloside transferase. Phosphoryloside transferase. Alright. Now, that's how we can convert these particular purines into the purine nucleotides. So first, we only have the purine bases, the adenine and the guanine. The remaining are remnants. Now, we are going to involve the enzyme adenine phosphoryloside transferase, the hypoxanthine guanine phosphoryloside transferase, of which we said this is the most important enzyme in this particular reaction. So let us talk about the regulation. Now, adenine phosphoryloside transferase is going to be inhibited by the AMP. So when we have enough of the AMP in the body, when you synthesize adequate amount of the adenosine monophosphate in the body, to mean it's going to have a negative feedback on the adenine phosphoryloside transferase, so that we cannot form the nucleotide from the adenine. So we're going to have a negative effect. We're going to inhibit. So AMP is going to inhibit that one. Then this particular enzyme, the hypoxanthine guanine phosphoryloside transferase, is going to be inhibited by GMP and also the IMP. Because remember, this enzyme catalyzes both reactions. So IMP. Alright, those are the regulations for the salvage pathway. Then, this enzyme here, just as I told you, is very important, and therefore, its deficiency or its defect, so that it doesn't function, can lead to a condition called Lesch-Nyhan syndrome. The disease or a disorder called Lesch-Nyhan syndrome. Alright, that shows how important this particular enzyme is, the Lesch-Nyhan syndrome. Alright, guys, that's very much important. That's all about the salvage pathway. It's not such complicated like the novel pathway, because we're just going to recycle, right? We're just going to recycle this particular bases from the nucleotides so that you can make them available to the body. Remember, I told you that nucleotides are very, very much essential. They run a series of physiological functions in the body. The energy, the signaling molecules, the components of the enzyme factors like the CoA, the NAD plus, everything. So that is it for this video, guys. If you found this video helpful, please make sure you subscribe to my YouTube channel, hit that like button, and comment in the comment section. Let's meet in our next video.